Okay, I'm on one of our other experimental streams, and what we're doing today is a census of the guppy population. What you're looking at here is the upstream limit to our experimental reach. Above where you see the end of that tape is a waterfall. That waterfall, we assume, would be a very effective barrier to the upstream dispersal of guppies, and that's the top of our introduction stretch. And then the introduction site extends downstream to about 30 or 40 meters beyond where we are right now. And what we do once a month, originally we introduced guppies into some of these pools further upstream. And the guppies that we introduced were caught in a high predation locality as juveniles. We then reared them to maturity in the laboratory and mated them in the laboratory. Then everybody was tattooed so that we could recognize them as an individual, and then we pulled 2 scales so that we could genotype them. And the nice thing about the scale pulling is that we only need about 4 or 5% of the DNA that we get from the 2 scales to genotype them, and the rest of it is archived for future reference, because ultimately what we're going to have are pedigrees. We're going to have populations that grow and adapt to this new environment, and each new baby that is recruited into the environment is going to be collected and marked as an individual, and we're going to collect some scales so that we can define who the mother and the father were. But it also means that we'll have an archive of DNA from every individual, or nearly every individual, in 4 replicate evolving populations. Before I say any more, it's good to look at what the structure of the streams are. These streams are built like a staircase in that they're divided up into discrete pools and riffles. The pool is what you're looking at right in front of you. That's an area where the stream is relatively level and there isn't much current. If you were to look at current inside that pool region, very often it would be eddies. It would be going in swirls, and it would be sometimes flowing upstream, sometimes downstream. But at the top of the stream, you see a rocky step, and that's where the water is flowing more quickly and in a straight direction. And then further up is another pool, then there will be another riffle and another pool. So as I said, the stream is like a staircase. We have pools being the stair and riffles being the vertical juncture between each successive stair. So guppies like to hang out in pools, and they're not inclined to move from one pool to the next, to move through riffles, but sometimes they do. And so what we do when we collect our guppies is we run this tape measure down, and each time we collect the guppies, we record where they come from, and the guppies in each pool are kept in a separate container, so that after we take them back to the lab and measure and mark them, we then return them to the pool where we caught them. And so we maintain the sort of social structure that they had inside the pool, because the guppies are very social. They know each other and they know their environment. And so we keep the fish together and we return them to the environment that they came from. But if you do this month after month with known fish, and if you record each time where the fish came from, it also means that you have a record of individual movement. And so we'll know a lot about movement up and down from pool to pool. Pools are pretty stable. Not too many move in and out of a pool at any given time. We found for a 2-week period that on average 5% of them will emigrate. But over time there will be mixture of individuals from pools. And so we're going to be seeing a lot about what that process is like, just as a byproduct of the other data that we're collecting. And we'll be able to see whether or not movement from one pool to the next is, for example, a function of how that individual was doing, or what the social environment was that that individual was experiencing. That's just a byproduct that you can get from the kind of work that we're doing. But the main point is that what this means is that each month we come as a team and we have to collect very systematically in each of these pools and in each of the riffles between the pools, so that we can find every animal that's present in the population. We actually can get a statistical estimate of the probability that we caught an animal on any single census if it was alive, and then we also can get estimates of mortality rates. The way we do that is we look for gaps in the recapture record. So for example, if we caught an animal on month 1 and didn't see it in month 2, but then did see it in month 3, what that means is that in month 2 it was alive but it wasn't caught. And it's those gaps in the recapture record that actually give us the information on how good we are at catching the animals if they were really there. In a really good month, we've had recapture probabilities of as high as 98%. That means we had a 98% probability of seeing that individual on that particular month when we did our census. On other months, if it's rainy and the waters are high and they're murky and we can't see them very well, then the probability of capture in a single month may be only on the order of 65%. But then we can also use those numbers to get some idea of what the mortality rate is and whether or not different groups of fish are more likely to die in a given interval of time. Now, we don't begin to mark the animals until they're about 14 millimeters long. When we see smaller guppies, the babies that are moving around in the pools, we leave them where they are, because what I learned in my work earlier is that those little ones are very fragile, that if you pick them up and bring them back and count them and then bring them back to the river, that there's maybe a 10% chance that they aren't going to get back alive, just because however careful you are, an animal that is that small is delicate and is going to be prone to injury. But the other thing is that until they're about 14 millimeters long, we can't safely mark them. Now, we also know from our earlier work that the probability of survival from birth to 14 millimeters may only be on the order of about 80% or 90%. So it means that a lot of selection has happened before they reach that size class. But we can learn about what natural selection is doing by comparing those new recruits, the ones that were born in the river and grew up there, with what their parents were like or what the previous generation was like. And so we can make inferences about at least what natural selection has been doing to them over the course of time.